Yeah, we will start in few seconds, please. Now we can start, I think so. Hello, everyone. I welcome you all for the webinar on cancer strategies, which is brought to you by the Biomedical team. Today, we have one of the leading scientists in oncology, Dr. Sandeep Singh, who is currently working as an associate professor in National Institute of Biomedical Genomics in West Bengal, India. I thank Dr. Sandeep Singh for accepting my invitation and making your George's presence over here with us today. Our speaker will reach you all with a talk on spontaneous emergence of a non-convertible cell state from a putative stem-like cancer cells in oral cancer. And his talk will be followed by question and answer session. So the participants are kindly asked to put your questions in the chat box of YouTube. And I am much delighted to introduce our today's speaker, Dr. Sandeep Singh, who is currently working as Associate Professor in the National Institute of Biomedical Genomics, West Bengal, India. He was a postdoctoral fellow in Huxley Motive Center, Cancer Center and Research Institute in USA. He pursued his doctorate in PhD Cancer Biology in National Center for Cell Science in Pune in India. He did his Masters of Science in Biotechnology from Himachal Pradesh University in India. Dr. Sandeep Singh has a broad background in cancer biology with specific training and expertise in stem cell biology of cancer, tumor microenvironment, and cellular signaling. His research interest includes mechanistic understanding of genetic and epigenetic heterogeneity associated with aggressive tumor behavior. Doctor Sandeep Singh has contributed to understand the role of stem-like cancer cells in aggressive cancer behavior. He returned back to his home country and achieved a highly competitive grant funded by Wellcome Trust India DPT India Alliance. With the help of this grant, he established the necessary infrastructure and initiated a project to define mechanism of heterogeneity among stem-like cancer cells and its functional interaction with the niche. Doctor Sandeep Singh is carrying out in-depth investigations on oral tumor biology and currently emphasizing on gingival cancer, oral cancer. His work has recently revealed the surprising diversity among fibroblasts within the microenvironment of tumor as of oral tumor as responsible factor for differential stem cells of oral cancer cells. Studies suggest that myofibroblast differentiation of stromal microblasts provide more conducive environment for aggressive cell renewal of oral cancer stem cells through differential expression of certain genes. Dr. Sandeep Singh was awarded as Intermediate Fellow from Wellcome Trust TBT India Alliance, India. Such an eminent resource person is with us in our Biomix platform today. With this introduction, I request Dr. Sandeep Singh for his talk.
Thank you very much, Partha. It's my pleasure. It's privilege to be again associated with Biomix. Thanks for again offering this opportunity to come back and introduce to you the current work which is going on. So today I'm going to share with you something which is still unpublished, all new, and looking forward to get responses and questions regarding this work. So this is going to be a little bit technical, and I have kept it as simple as possible for the general audience also. So looking forward to hear from the audience.
Yeah,
I'm
sure, sure.
sharing my slide.
Yeah.
I hope it is all visible.
Yeah, it's all right, sir. You can
Okay,
proceed.
so I think it's changing, right?
Yeah, it's moving, sir.
So thanks again, and thanks for your elaborated introduction. So I don't need to introduce myself anymore. I just like to introduce my institute from where I'm coming. This is National Institute of Biomedical Genomics, and uh, this is the website nibmg.ac.in. So everyone who wants to do cutting edge research in India are welcome to visit NIBMG. They should get all the information about their training, which is offered at NIBMG in the form of PhD or integrated PhD for the graduate students. as well as short-term training for all those who has training in their curriculum. We are on Facebook, we are on Twitter, so we are well connected. So you can approach us. And with this, uh, I would like to tell you that my interest is to understand Indian oral cancer. Okay, so what is Indian oral cancer? We are very much used to of seeing these all around us, right? All tobacco products are so much available in market. Even our kids can go and buy these products from market without any law or regulation. So this is also advertised by our heroes to whom we follow, and that is what has made this problem a bigger problem, because we want to follow them, and based on the surrogate products of uh, tobacco associated chewing tobacco associated products. When we started to see each cancer more closely, we very quickly realized that not only the cancer is different between person to person, the cells within the tumor is actually different between the clones present in the cancer in the individual tumor. So the during the course of the cancer progression, the evolution may occur, and because of the evolution, there are subclones which actually comes up in the tumor, in the patient cancer, and then we see multiple clones of cancer cells which may be existing. So this is called intratumoral heterogeneity, right? There are some clones which will actually spread and become what we have actually understood very in detail uh, by the genomic study of cancer, right? So this is all genomically heterogeneous clones of cancer cells which may exist within the patient who is getting treatment. So some clones may be raised into treatment, and they again grow back when the treatment is over. Now, with more advancement in the technology, we can actually go and look at each of these cells individually. By doing that, we can actually find whether the cells are all the cells which is sampled is same or it is different. So if you take the biopsy of the cancer cells from different region of the tumor, you will see that there are different at the level of genomic changes, as well as they are different on the transcriptomic and protein expression changes. So this is where we are right now, where we are actually acknowledging the phenotypic heterogeneity in genetically field is right now. So we have moved from patient to patient variation, and now we are moving towards the transcriptomic, the phenotypic heterogeneity among genetically very same cell. So this is where this is the complete picture of aggressive cancer. And what is the biology of this phenotypic or transcriptomic heterogeneity among genetically similar cell? We will not be able to come to the medicine which is precisely going to treat cancer. So this is where the field is moving, and the field is called the precision medicine. I'm not going to do the detail more on this. I would just like to tell you very briefly what we have come up with in the past 4 or 5 years of this research. So we have tried to understand how uh, non-genetic heterogeneity can arise in oral cancer, okay, in gingivobuccal oral cancer. So this is a very famous picture of the developmental aspect, developmental biology. So if I relate you with respect to something which you can immediately link off. So we all are genetically the cells which is there in our body, they are all genetically similar, right? Our eyes have the same genetic makeup as our lung cells are, but our eyes have different function. The genetically similar, there is phenotypic and transcriptomic heterogeneity, and this is what
So now you can start, I think so. It's off,
Okay.
right?
Yeah, there was some technical issue, so I'll again explain this. So as the cells are changing the state, they take the decision of going into which differentiation path they need to take. And with the advancement of the technology now, with the advancement in the knowledge, not the technology, technology is always advancing our knowledge. So with the advancement of the knowledge, we now know that these cells, the differentiated cells, can actually trans differentiate, and they can become different cell type, or it can actually go back into the state which is called de-differentiation. So these states are quickly switching depending on the need, depending on the stress condition, depending on the homeostasis requirement of the tissue, and this is what is the current understanding. So if in the tissue we are seeing these stem cell behavior uh, giving the non-genetic heterogeneity, does it happen in cancer? So the cancer is arising in the tissue. The tissue has the resident stem cell, which is getting the cues, the signals from the surrounding cells, which is actually making its home, the niche. The cells, which is stem cell, they self renew, and they generate differentiated population of the same genetic makeup. In cancer, these resident stem cells, they acquire mutation. They become cancer stem cells, and the cancer stem cells can actually generate accumulate more and more mutation and become more aggressive. So cancer stem cells actually act as the unit of evolution. So there may be different diversity of cancer stem cells based on the genomic makeup, but each genomically distinct cell will actually generate the bulk population of cancer cells with that genetic makeup. So there is non-genetic heterogeneity which is arising from cancer stem cells, which may form the heterogeneous tumor. So cancer stem cell theory provides a conceptual framework to describe aggressive cancer behavior. And cancer stem cells are the subset of cancer cells with stem cell-like ability for long-term regeneration through self-renewal and differentiation. Therefore, these are suggested to be the responsible for tumor initiation, progression, metastasis, resistance therapy, and recurrence. I will suggest that all who are interested in understanding what are cancer stem cells, you should please follow my previous talk given on the Biomics platform, and there I have talked about the basic biology of cancer stem cells, the research which is basically happening right now. So with this concept, I wanted to understand whether the gingivobuccal heterogeneity is driven by non-genetic mechanism also. The genetic heterogeneity is already understood. We have sequenced. We know that there are driver genes, there are passenger genes, but whether the non-genetic heterogeneity exists in cancer and whether it has any relevance to the clinic. For that, I collaborated with Tata Medical Center, which is in Kolkata, and Dr. Arun, Dr. Rajiv. They are the surgical oncologists, very, very motivated to do research. And we actually clubbed together, we made the team, and I started to collect tumor samples, which is coming from the gingivobuccal region. We collect a lot of samples, and as you know, this is coming from the oral cavity. The oral cavity, which is so open to the environment, the cancer is growing. You cannot clean the mouth. There is so much fungus. There is so much bacterial, microbial growth, and that was the major challenge to do anything with these patient sample. So this is the number, and we ended up with developing our own cell line. So this is unique. This is novel because in the world there are scarcity of a gingivobuccal oral cancer model, and especially these are the cell lines which is established from India, from the buccal mucosa, which is the most common site of getting oral cancer in Indian population, and this is the only cell line. There are no other cell line available in the world to work on buccal mucosal oral cavity cancer. So this is the method. I'm not going to go into much detail. So we collect the tissue, we process it, we put it in the culture media, the cells start to grow. And if they are not contaminated, if there is no outgrowth of the fibroblast, we get to see some epithelial cells growing. These epithelial cells are not transformed artificially. They are having its own genomic makeup, and because of the mutation, they actually generate as a cell line. We can grow them in 3D colonies. We have demonstrated that they are sensitive to cancer stem cell, anti-cancer stem cell therapy. They show very unique developmental signatures, and uh, they are having unique genomic makeup. So we took these cell lines and we asked whether these cell lines can be used as a gingival oral cancer model. So at NIBMG, we have International Cancer Genome Consortium project, which is going on understanding the genomic makeup of gingival oral cancer in India. So their data was already published, and we have already shown that what are the mutations which are required for people to get gingival oral cancer. So we mapped our cell line to the data which is available, and we found that yes, the driver gene which actually is required for driving the cancer of gingival oral origin, our cell line was having this. Okay, so this, you can see the CAF, the cancer associated fibroblast, is free of any mutation, but the cancer cell has a lot of mutation. These are the somatic mutation on the single nucleotide polymorphism, and these are the genomic, the long reason alterations, which is gene amplification, gene deletion. So you can see that the two cell line which we have developed, we have named them as GBC, gingival carcinoma 02 and 035, based on the number of the patient which we have actually given the ID. And you can see that the GBC02, while it has very fewer mutation, it has more of copy number alterations. So these are the unique cell lines. We have already published this. It is still in press. It is oral oncology, and you can follow the findings which we have already reported in this. So once the cell line, the cell culture model was established, we started to ask our question. The research focus of the lab is to understand functional heterogeneity in genetically identical cells. Functional heterogeneity in due to genetic alterations, and functional heterogeneity driven by tumor microenvironment. So these are three different ways the cancer cells can actually get functional heterogeneity, and this is what is our research focus is. For the time today, I'm going to focus mainly on the functional heterogeneity, which is driven in a genetically identical cells. I'm not going to touch on these other aspects. So, as I said, the non-genetic heterogeneity and non-genetic method of heterogeneity can arise from the stem cells. So what are the stem cells in oral cancer, which is already well understood? There are three markers. ALDH1, which is aldehyde dehydrogenase enzyme. It's a metabolic enzyme which is responsible for oxidation of aldehydes to carboxylic acid. The CD44 is a cell surface marker which binds to hyaluronan protein in the matrix, the extracellular matrix, and it helps the cells to migrate by adhering to the matrix. CD24 is a glycoprotein, which is a cell-cell adhesion protein, and it also helps in migration. So these are the three markers which are very well studied for defining cancer stem cell like behavior in head and neck cancer. Okay, so these are some references which I have put here, and this paper is from India. It is, but the cell line is all developed from other places, and what it is telling in very brief is that in the bulk cancer cell, the CD44 positive cells will have a small subpopulation which is ALDH1 positive. So this small population of ALDH1 positive can be called as cancer stem cells. So what the definition should be is CD44 positive, CD24 negative, ALDH high. So this is the definition which is actually is summarized from these studies. And it is not the only cancer where the CD44, CD24, and ALDH is famous. These three markers are also shown to have cancer stem cell property if the cell has these markers for variety of solid tumors. So we took these as our benchmark to go ahead and start working. I will just very briefly tell you what is how do we actually find cells with high aldehyde dehydrogenase activity without disturbing the cell viability. This is by aldafluor assay. This is available through stem cells. It's a proprietary item of stem cells, and now Sigma has come up with its own surrogate of this method. So there also we can actually identify cells with high aldehyde dehydrogenase activity. So very briefly, what we do is we take the cell population, the mixed population, we put this reagent, which is the substrate for aldehyde dehydrogenase. Okay, this is body P substrate, which is going to become more fluorescent if the cells are ALDH positive. But if the cells are not ALDH positive, then the substrate is not fluorescent, and you can identify them. How do you know that the cells are LDH positive or not? So they provide a very specific inhibitor of ALDH, which is called DEAB. The full form is written here. So if you add DAB in the cell, which is even if the cells are positive, they will not be able to convert the dye. So the cells will not be fluorescent. So this is the case. You take a tube where you have the cells which may have the ALDH high activity positive cells. You put DAB, and then you make a gate saying that any cell which is coming in this gate because of the high aldehyde dehydrogenase activity, it should come here because this is my background. Okay, so now the cells without DAB start to produce more of this fluorescent molecule, and you start to see them in a different gate. Okay, so this is the basis of finding the high aldehyde dehydrogenase activity positive cells through flow cytometry. So now the CD24, CD44 are the cell surface marker. You can use antibody of that, and you can use this metabolic labeling by this reagent, and you can find the LDH positive cells through flow cytometry. So in this study, we have used a panel of cell line, and you can see that these are having a different genetic makeup. The 3 cell line which we have used is generated from buccal mucosa. We obtained these 4 cell lines. So I already told about the origin of these 2 cell line in our lab. We obtained these 4 cell lines from University of Pittsburgh. Okay, so this is Caucasian origin. This is the Indian origin. The 3 of them are actually generated from retromolar trigon. This is the behind your molar teeth, the region of the gum. So these are originated from there. So basically, this is the model for gingivobuccal oral cancer, and we have relied on these cell culture models. Now, the first aim was to understand how these cells are for the expression of CD24, CD44, and ALDH. We combined all 3 together. Okay, so just focus on one of these, not in all of them. Just focus one of these. This is where you have CD44, and this is where you have CD24. So as the cell, as you see, as you go high on this axis, you have higher expression of CD24. So by combining CD44 and 24, we could get very specific 2 population. CD24 negative, but CD44 positive, because it is very high for CD44, and the CD24 positive, CD44 positive. So in CD44 positive cells, there are 24 positive and 24 negative population. But now you can actually see what is the aldafluor positivity in these 2 population if by flow cytometry. So you can make the gate here, and then again you can ask whether the aldafluor positive cells are present here or here. So the aldafluor positive cells were actually present in this 24 negative population at 17, say here 17% frequency. But at the same time, when it was present in this 24 negative cells, which is CD44 positive, CD24 negative, ALDH high, the putative stem cells, which is already reported in the literature, we got the ALDH high cells even in the CD24 positive cells. So this was the beginning of this project, because the putative stem cells were already reported to be present in this population, but we were seeing another ALDH high population in CD24 positive cells, and that was not the case for one cell line. It was the case for across the cell line, different cell lines. We actually had a cell line which is only CD24 positive. There is no CD24 negative cells. They are all CD44 positive, but there is no CD24 negative. It is completely CD24 positive population. And the elder positive cells were there. Okay, so the elder positive cells are maintained in CD24 positive and negative population, and this is what the summary here. All cells were CD44 high. ALDH high cells were present in both CD24 low and CD24 high compartment, and ALDH high cells were less frequent in all tested cell line. Okay, this is the unique. This was the common theme across the cell line, that the cells which are ALDH high, whether it is coming from 24 positive or 24 negative, they are always maintained itself in a lesser frequency. So was this the case only for the cancer cell line, which is already having so much of may have so much of perturbation because of the culture conditions, or it is possible to have to see this in the tumor samples also? So this is the summary of the 7 sample which we have generated data, and we found that there is ALDH high population is in about around 5%. And when you see whether the CD24 positive and 44 expression in ALDH high cells, you are seeing that the cells are very highly most frequently present in CD24 low, CD44 high compartment, which is already reported in the literature that the cancer stem cells actually enrich in this CD24 low, CD44 high population. So this is anticipated, but at the same time we are seeing this less frequent population even in CD24 positive cells. So in the literature, if you search, you will not find much of the data. People have not talked about the property of these cells. Okay, so this was very interesting to us, and we took the challenge to actually try to understand what are these cells and why they are also present, why these cells are maintained in the tumor. So the questions we asked is, does CD24 high ALDH high cells also possess stem cells in addition to the putative stem cell marker already suggested in the literature? And what is the cellular relationship between these subpopulations? And what is the significance behind maintaining these heterogeneous subpopulation in oral cancer? Okay. So we wanted to take a very unbiased approach, because this is something which we were going to propose, something which is not very well appreciated or very well acknowledged in the literature. So we wanted to take an unbiased approach. So we worked on all 4 population at the same time. Okay. So the first argument which anybody will have to us, or you will be having to me, that the cells are maintained at very different frequency. So the cells can be maintained at very different frequency because it may have a different proliferation rate. So first thing which we asked is whether these 4 population have very different proliferation rate or not. So this can be done by dye dilution assay. We can actually stain all the population at the same time with the same amount of dye. So it is always dark. But when the cells divide, they are going to half the dye between 2 cells. And this is how the dye will actually getting diluted. And then you can see whether which population has divided maximum and which population has not divided. When we did that, and we followed this division for 4 days, we found that all 4 population, whether it is coming from CD24 high ALDH high or CD24 low ALDH low, CD24 high ALDH high or CD24 high ALDH low, they all are proliferating, they are all dividing. There are some differences, but between the biological samples, we don't see any significant difference between these four population. So if you have noticed that we have actually followed a very specific color code. The putative stem cell is given a red color. The CD24 low ALDH low is given orange. The 24 high ALDH high is given green, and that CD24 high ALDH low is given the blue color. So for your ease, these color codes are present in all the slides. So you can please follow this. Otherwise, you will miss somewhere. So because all the cells were proliferating at the same rate, we ask the question whether they have equal colony forming ability. So this is another property of the stem cell, which people do commonly in the in vitro assays, that you plate the cells in very low frequency, well apart from each other, and if they have this long-term long-term repopulation ability, they will actually generate colonies. And all four subtypes were actually able to form the colonies. So it became very interesting. So we started to ask more complicated questions. The second question which we asked is, if a particular subpopulation has the stem cell property, then it should show totipotency. Means if we are working on the four population, it should generate all four population. And if the cell has low totipotency, if it is differentiated, then it will remain differentiated. So with this idea, we sorted these four population, and then we gave 10 days for the colonies to generate, just like I showed in the previous experiment. All the four population were generated colony. We collected the colonies and rechecked their the repopulation of these cells by same assays. So when the red cell is separated, you can see the sorting here. The red cell is separated. Almost 99% cells are red cells. But when it generated all four population: red, orange, green, and blue. The orange is separated. The orange could actually form orange and blue, not all four. The green is separated. The green could form green and blue maximum. There is some spillage to the orange. And when the blue is separated, the blue is forming mostly blue. So this was the property which we actually found in the population across the cell line. So this is with different cell lines. Okay. So GBC02, 070, I have already shown you they are very genomically very different. So even where the genomically very different population, their repopulation ability is fixed. Means the red population is dividing, it is forming all the three population. But when the green cell is dividing, it is forming green and blue. Okay, exactly the same way as has been done for us during the during our development. So any organism, say human, we all are very different from each other. We are genetically very different, but we develop eyes, two eyes, we develop hands, two hands. There is overall phenotypic similarity is maintained among genetically very distinct populations. So this is happening because of our cells have undergone a repopulation memory to generate a very specific cell type after the division. And this is what is actually recapitulated by these cancer cells, which are genetically very perturbed, but they have actually formed the population, and this is across the mutation type which we have studied. So we asked our friend Dr. Mohit Jolly from IISc Bangalore, because he has the unique ability to generate mathematical model, and we were seeing that something is happening across the genotype, across the cell line, and we can generate numbers, right? We can generate frequency of, we are putting 99% red population, we are getting only 10% at the end. So we gave these numbers to him. So what he generated a trajectory plot for each phenotype. So you just follow here. So what he's suggesting in this plot is, when the population starts with 100% red cell, And this is the x-axis where you can follow the time. At time 0, when the 100% cells were red, with time the red population comes down and the orange population starts to go up. It is followed by the blue population to come up. Okay, so the biological data which we have given to him, he could generate a mathematical model. He could shuffle those percentages which we have given to him to achieve the steady state, the end point, that this should be the frequency of our population at the end, because we have found this from multiple experiment by after passaging, that the red population is always maintained at less, the green population is always maintained less, but the blue population is always maximum. So that is the steady state, that's the end state. So can he propose a mathematical model for that, a formula for that for us? And using this formula, he has plotted this graph. So when you start with red, you end up with blue. When you start with orange, you end up with blue. You start with green, you end up with blue. And when you start with blue, you remain blue. So it says that the red and our biological data combined suggests that the red population is the most frequent, the most pluripotent, and the blue population is least pluripotent. So the after generating this relationship between the population, that the one population is giving rise to other populations, we wanted to understand whether they are molecularly related. Okay, for that we went ahead and generated uh, the RNA sequencing data. So again, we have the mixed population in the plate. We can separate them by sorting. We can generate RNA sequence data from each of these population. This is our RNA sequencing data. So this is these are the gene expressed, and we have a specific number for their expression, right? So with this number, we can actually ask our bioinformatician friends, and Kavya has actually learned these things in the lab, and she can do very efficiently now, that she has put this data into the computer and asked whether it generates a pattern. So here we have given a list of genes which is very well studied in stem cell biology, right? So what I'm showing you here is there are some genes which are uniquely expressed in red population. It is not expressed in other population, but you come down to this list here, you start to see that now orange population has also started to generate this, right? So now you can actually visualize that these populations are getting related, right? Now you come down here, you see that the red population is having less expression, whereas the other population has started to express these protein at higher. So this is generating a pattern. Now you can ask better questions by doing the differential expression. So she generated a differential expression data for each of this population with each other, right? The blue versus green, the orange versus blue, and so far and so on. So we have 4 populations, 16 permutation combination can be generated. She generated 16 differentially expressed gene signatures, and this is the summary of the data. It suggests that the blue and the green population are very similar to each other at the transcriptome level. It has only 4 upregulated and 17 downregulated. So very close to each other. The transcriptome is very close. The transcription state of the blue and green is very similar. Orange and blue, almost 300. Red and blue is more, right? Red and green is more. So obviously blue and green very similar. So red and green and red and blue will be very different. And red and orange is again coming to closer, because the differentially expressed gene is actually almost half as compared to red and blue. So you can actually plot this in the hierarchical clustering, and with this you can actually generate a map that which population is close to which population. By doing this, what Kavya and from Mohit's lab, Priyanka has come up with this idea that the red is very unique, followed by orange, followed by green, and green and blue is very similar. So again, it is giving us the similar pattern what we have actually got from our repopulation analysis. The red is giving rise to blue and green, and then blue remains uh, as blue type. So while we were making sense of these observations, we came across a paper which is published by Ophir Klein lab, and he published this data in cell stem cells. So with the mouse model, his question was that how come our buccal mucosa or the tongue or the entire oral cavity, this paper is about buccal mucosa, but the bigger question is the oral cavity. uh, we have so much of wear and tear while eating, uh, while cleaning our teeth. We have so much of wear and tear, but our oral cavity heals itself very fast. So he took the example of the buccal mucosa of the mouse and generated single cell sequence RNA sequencing data from the buccal mucosa. Okay, so uh, so he focused on the basal layer, which is actually the niche for basal, the squamous cell uh, epithelium of the buccal mucosa. And the if you take the basal cell here and you do a single cell RNA sequencing, what he found is that the stem cell like property, the signature of stem cell, is not only maintained in cytokeratin 14 positive putative basal stem cells, but actually it is distributed in all the population, which is coming out from there and ready to get differentiated. Okay, so what is the significance of this? The significance of this is that the cells are ready to renew the epidermal layer if required. So as soon as there is damage, all these cells which have poised level of stemness, they will immediately get differentiated and repair the tissue. So that's the reason why the oral cavity, especially the buccal region and the gum region, because the canine of the mouse always grow, right? The gum and the buccal region is so fast renewing after the damage. This is what they have proposed. What Kavya did is she took the gene expression signature, which is present, which is actually proposed by them for these basal stem cell and differentiating population. Okay. And we already had our population based RNA-seq data. She overlapped that. And you can see the difference here. The genes which are upregulated in red as compared to green, you see that there is higher overlap with the stem cell signature as compared to the differentiating signature. Okay, the red population is closer to the basal stem cell. This is what is coming by overlapping the mouse RNA-seq data and the cell line RNA-seq data. It is suggestive that the red population is closer to the stem cell population. But very, very interestingly, you come to green population, the orange population, the blue population. What you start to see is that the green population or any of these population has maintained its similarity with the stem cell population. At the same time, it has started to express genes which are associated with differentiation. It is this was the case with the green, the orange, as well as the blue population. So the cells are differentiating. It is showing that cells are differentiating, but while differentiating, they are still showing its similarity with the stem cell population. Exactly what Ofer Oakland has published in his cell stem cell paper in 2019, that the buccal basal buccal basal layer is maintaining putative stem cells in differentiating population in the buccal layer and the basal layer. So Kavya next did this, that she did the gene set enrichment analysis for the genes which is upregulated in this green population. Okay, so the gene set enrichment analysis clearly suggested that the cells are differentiating, because the cells are having keratinization, conified in envelope epithelium kind of signature which is coming up from this green population. So we ask the question, if the cells are showing similarity with the stem cell population, so does it maintain stemness also? So for that, again, the cells were sorted from the cell line, the four populations, and the basic stem cell markers like SOX2, OCT4, nanoxemic, KL4, SOX9. We performed the RT-PCR for these markers, and the RT-PCR results clearly suggested that the red population has higher expression of SOX2, but the other populations have higher expression of some of other stem cell markers. And at the same time, all the population are expressing the basal layer cytokeratin, just exactly the basal layer present in the stem cell present in the basal layer in the mouse buccal mucosa. We are showing that even in a developed cancer, the cancer cells actually maintain the basal cytokeratin level in all these four populations, which are differentiating, as evident by the other differentiating cytokeratin marker. So the cells are differentiating, at the same time they are maintaining stemness. This is our RT-PCR data. Biomedically it was proven. What Priyanka and Kavya did is that they did single sample GSE analysis to see whether our gene signatures, the genes which are expressed in our four population, is maintaining its similarity with the embryonic stem cells or the SOX2 target in the mammalian stem cells, the adult stem cells. So there was no significant difference between these four populations. So it suggests that these four populations are maintaining stem cell within it. And this is again, we did this similarity assay with the signature which is Klein Lab has given. So they have given the transcription factor which were associated with basal layer, the basal differentiating cells, and basal cycling cells. So all four populations showed no difference between these four populations, these three signatures, suggesting that the cells are differentiating, at the same time maintaining the transcription factor for the stemness, differentiation, as well as they can cycle. And that is what is actually correlating with our first result, that there is no proliferation difference between these four populations. So we did some functional assays to understand whether the stemness is really being can be shown here. This is by sphere formation assay. You can see that we can actually plate as less as 5 cells, and from 5 cells to 50 cells per well is plated to check whether the cells are forming spheres or not. So this kind of dilution assay will actually suggest us the frequency of the stem cell which may present. So you can see that the red population has started to form the sphere very early, means at very low frequency of cells plated. You can see that the sphere formation ability is seen, and the blue population is the least stem cell forming, the sphere forming, suggesting that the least accumulation of the stem cells here. These are the morphology of the spheres. We can actually passage these spheres. We can see that the primary and the secondary spheres can be generated from these population which is developing the sphere. Most importantly, what Kavya has generated is the estimate of the stem cell frequency positive cells, the stem cell property positive cells in these four populations. Okay, so now you can see with this number, we can actually estimate this number, and it says that among 100 cells, among 3 cells, 1 cell will be sphere forming in the red population. 6 cells, 1 cell will be sphere forming in the orange population. 1 cell will be sphere forming from the 11 cells in the green population, and 1 cell will be sphere forming from 24 cells in the blue population. So this is clearly suggesting that even though there is differentiation, which you can see here, the cells are differentiating because their sphere forming efficiency is going down, but the sphere forming efficiency is maintained, the stemness is maintained. So we cannot ignore any of these population when we have to study the stemness in oral cancer. This is what in brief I want to tell. uh, so we went ahead and asked whether our RNA-seq data can generate any clinical significance and suggest the relevance of this heterogeneity which we have found in these four population. Again, took the help of Mohit, and Priyanka has generated this data. What we have done here is that we have generated unique genes which are present in red, orange, green, and blue. Not differentially expressed gene, but the unique gene. Unique genes which are expressed in red, unique genes which are expressed in all other population. And these unique gene signatures are matched with the TCGA data, the consortium data, which is having the data from 500 more than 500 head and neck cancer patients, and their RNA sequencing is done, and their survival data is already available to the public, who can actually generate data from there. So what Priyanka has found is that the red population, the unique genes which are expressed only in the red population, is showing a better prognosis. The putative stem cell signature is giving a better prognosis. But if the patients have lost the signature of the red gene, they start to behave poor. The tumor has started behaving poorly in the clinic. They are more aggressive than those patients who is showing the signature of the putative stem cells. So what is the meaning of that? The meaning of that is that if the stem cells are there and the cells are maintaining those stem cells, the signature of the red population, and the red population is not forming other four population, the cancer may be better. But if the red cell is there which can actually generate other population, then the cancer become worse. And why does it become worse? Because of the enrichment of arachidonic acid metabolic process. Okay. What is the relevance? We are still trying to understand. We are trying to model this. This is the arachidonic acid metabolic process is published by ICGC, the ICGC India project, in Nature Communication paper from NIBMG, where the mutation in arachidonic acid metabolic process, metabolism, the genes which are associated with arachidonic acid metabolism, if the patients show mutation, and by the way, this mutation is the loss of function mutation. So the function is lost. So if they are having mutation, they survive better, and if they are not having mutation, they survive poor. So arachidonic acid metabolism pathway is associated earlier at the mutation level. Now we are showing at the transcription level. The non-genetic heterogeneity is also given aggressive behavior because of this metabolic process. So in conclusion, what we have proposed is that there is heterogeneity in the tumor, and so there are two different states, that two different levels of heterogeneity we are talking about: the metabolic heterogeneity and cell type differences. So here the cells are changing from one state to other state, the 24 negative to 24 positive. This conversion is non-convertible, but the metabolic state is convertible. And this is what we have found, that the orange population can become red, the blue population can become green, but the red population once become green, the green cannot go back and become red. And this is the origin. This is where the heterogeneity is coming. Once the red has made green, the cancer is aggressive and the patients are behaving poorly in the clinic. The take-home message is, unexpectedly, we found that oral stem-like cancer cells were very versatile and less compartmentalized as has been proposed previously. Cells acquire different functional state by conserved process of cell division. This pattern of cellular plasticity appeared to be not affected by genomic differences between the cancer cells. And heterogeneity in oral cancer is driven by cells with diverse transcriptomic states and can be associated with aggressive behavior. With this, I would like to say thanks to my collaborators and my friends at NIBMG. The most of the RNA sequencing data is all done with the help of Nidhan. The genomic sequencing data is done with the help of Dr. Arindam. Dr. Samsiddhi is helping us with the statistical interpretation of the data. Dr. Molina, Dr. Kartiki are dear friends. And without the help of Dr. Arun, Dr. Rajiv, this project would have not kicked off. This is all help of them, and I'm immensely thankful to the help provided by the patients for their consent to give their valuable samples. Mohit for the data which I have shown already to you, and the entire project is funded by Welcome Trust DBT India Alliance, and the infrastructure provided by NIBMG. The data which I have shown you here is all produced by Kavya, and the other lab members are also helping her with some aspects. And Ankit has already passed out, and he is doing postdoc in Sweden. So with that, I will thank and take questions, Partha, which you might be having. And it was nice presenting my data to you.
Yeah. Yeah, thank you so much, sir, for your excellent presentation. And the way you presented towards your results is obviously showing that your results has been correlated with mathematical modeling as well as in uh, as well as one. Hello. Yes. Am I audible, sir?
Yes, yes, Partha.
Yeah, sorry for those technical issues. And I congratulate on your lecture today, and you are presenting your results based on the mathematical modeling, and you combined your results with bioinformatics tools. And it was so interesting lecture, sir. We have some few questions for you, and I hope you can take these questions from our
Sure, it's my
audience
pleasure.
as well. Yeah.
Sure.
Um, the first question to you is, Dave, we have seen CD24 cells in four different populations reacting on ALDH1. And can we know what about CD44 in four different population cells with ALDH1?
Right. So the cell line which we had in our hands, they were all positive for CD44, uniformly positive for CD44. Okay, the all four populations were uniformly positive for CD44. But when you see patient data, we have CD44 negative population also, where the ALDH positivity is there. But unfortunately, the cell lines which we had, so the current research which should be done, or the future research which should be done, is to be able to actually generate cell lines or cell culture model from those CD44 negative population, which will actually give some more insight on the biology of fluorescence. So right now we don't have any answer for CD44 negative ALDH population.
Yeah, the next question is that in the four different population, we have concluded that stemness, differentiation, and proliferation rates are same, and there is no moderation in these properties. But when we go for expression of SOX2 level, even though we witnessed stemness and differentiation and proliferation rate are same, but when we compared with red, the putative stem cells, it shows high level in the expression of SOX2. What may be the reason for this?
Yeah. So this is what was the enigma, right? So we were seeing that somewhere it is very similar to each other, but somewhere it is very different. So we found that red population has higher SOX2. The other population has higher of SOX9 and other OC4 and nano OC4 and CMYC. So and then when we actually compared with the data set which is given by uh, Offer Oakland paper, we actually could generate the knowledge, the understanding that the red population is more closer to the stem cell population, the basal stem cell population, whereas the orange, green, and blue is more closer to the differentiating population, which is still of basal, which is still in the basal layer and can act as putative stem cell when there is a demand for tissue renewal. So this is where we are actually coming to that the stemness is maintained in all the four population. The cells are differentiating. The, with the differentiation, it's not like all the population, all 100% population of the cells with that phenotype has completely differentiated or is differentiated. The stemness is also maintained in some of these population, even though they have the phenotype, the marker expression of the differentiating population.
So, the next question to you is when we come across DEAB, which is the obviously inhibitor of this fluorescence, and is there any idea to artificially synthesize this DEAB as a inhibitor to inhibit the cancer stem cells so that we get rid of this?
Yes, if definitely DAB would have been a clinical drug, this company would have not given us for doing experiments, right? So I don't know how much stability it has in the body, but definitely similar molecules which can inhibit aldehyde dehydrogenase activity is already in a big research to develop the inhibitors for aldehyde dehydrogenase activity (LDH), but not DAB certainly.
Yes, sir. And when we speak about intra-tumor variations in a particular individual, it has we come across many different kind of phenotypical cells. Is there when we combine these tumor, which is in a particular individual, with the that of other existing individuals, is there any cells? It's very specific to a kind of individual and not resembling in other individuals, or a cell which is very specific and it is enormously generated in a particular individual where it is not evidenced with that of any other individuals, or a very particular a new kind of cell is being synthesized in a very particular individual where it is not evidenced in other individuals. Anything is being reported like this?
Um, so if I have understood the question, it was very big, first of all. So, but you know the intratumoral heterogeneity is being focused right now with our ability to see the tumor or the cancer at single cell resolution, right? So what we are actually understanding very quickly that the cancer has the tumor comes with different cell types, right? So right now it's a cell type. There are many different cells which are present in tumor microenvironment. So there is huge heterogeneity. and within that each population there are sub the states, different states of cells. So with this, a lot of data is getting generated, but how it is related to the biology, how many states are going to have similar function, these are the questions which needs to be answered for understanding the variation between the cells and its biological or clinical relevance. So those data is still we are waiting to be generated, but single cell genomics data have started to show us the state differences in among cancer cells within a particular tumor or across the patients.
Yeah, sure, sir. We have a few questions from our audience, and these questions are, I will read out the questions. Is the
Okay.
saliva of a particular person helpful in detecting a oral cancer in its starting stages? And also, is there any method by which we can make salivary amylase enzymes to defend it?
Mm, as per my knowledge, I have not come across this kind of study, but a lot of metabolism study is being done, is going up and going on, and people are showing that the metabolic proteins, metabolic agents which are present in saliva, how they are different from a healthy person to the patient, the precancerous, the cancerous, and so on. And with the metabolic differences, I don't know about salivary amylase, but the metabolic differences are suggesting some thoughtful things which can be followed up.
Yeah, so the next question is that the slides we have shown is these cells or monolayer or suspension methods is being cultured.
Right. So the pre-population assays which we have done, which I have shown, is all 2D. It's a 10-day assay. But at the same time, we have generated a repopulation data from suspension culture. We have generated the spheres and collected each clone, each of these spheres, and shown that how these clones are actually very similar when they are plated. So I have shown the data from 2D, but we have generated some data from the suspension culture also. But other RNA-seq data, the qRT-PCR, we have generated from suspension culture. The RNA-seq is generated from the 2D culture.
Yeah, the next question is that how can we use suspension culture for these experiments?
Okay, so if I will suggest, so there is a method, you have to follow the method, uh, but I will suggest that you go through the oral oncology paper which we have just published. So there we have given the detailed protocol for generating suspension culture for oral cancer from the patient derived oral cancer which we have generated. So you will see that how we have gone through the step by step to generate the suspension culture and use them for doing drug screening or RT-PCR or the transcriptome studies.
Sure, sir. The next question is that in an area of the oral cavity where squamous cells carcinoma has been detected, does surgical treatment involve removing the proliferative basal layer?
Yes. See, when the tumor has already generated, there is no basal layer remained, right? So this is the 3D tumor which is growing. The basal layer is remaining in our normal mucosa. So when you take the tumor out and you cut the section, you will see that multiple lobes of cancer is there, and each of the lobe has its own basal layer. So this histopathologically we can see, and based on that, the patient's cancer is actually divided into uh, moderately differentiated, poorly differentiated. This kind of differentiation which is given is based on how much it is looking similar to the normal mucosa which is present in the not healthy people. So ultimately, when the tumor is grown, the basal mucosa is not identifiable. It is all mixed.
The next question is that during cancer research, we consider one gene or trait at a time. how much would it be useful if we target multiple genes and treat using multiple drugs which are already studied?
Right. So everything what you do has a plus and minus, right? So the way the research has been done, the initiated with single gene. People work with SARC, people work with RB, people work with E2F, P53, and they thought that this is the target. But eventually, now we know that medicine is highly heterogeneous, and we are in the era of informatics, the big data. So we can generate the pan data and come up with the idea that what kind of pathways which should be targeted, not the gene, because the same gene may not be mutated or altered in the next patient, but may belong to the same pathway. So this is where we are right now. We are trying to understand which pathways are altered, and this is where is the field of precision medicine. We can segregate patient based on the pathway alterations. So this person belongs to MAP kinase pathway. This person belongs to P53 apoptotic pathway. So with that, we can actually identify, we can actually test those drugs which is already in the market, or find a novel drugs which will perturb that pathway. So this is where I believe that we should move.
Yeah, sure. So the next question to us is, there is a large data set available on gene data set in NCBI database. How it is that useful for research in India, as the data is of the people living in different countries and cancer might cause due to different reasons?
Yes, so this is why uh, the Department of Biotechnology, Government of India has initiated the programs to generate data which is India centric. So we have the data which is available in on internet, on databases which is developed in US and Europe. But how much it is relevant to Indian condition, right now we don't know unless we actually go and probe it. So at the same time, when Indian government has given funding for generating India-centric data, like ICGC India project, where the aim is to generate the mutation profile, the catalog of the mutation which is occurring in the gingivobuccal oral cancer in India. But at the same time, we need to go and check the data with the available European or US database data sets, and see how much we are similar, or whether we are very much different. So unless this is done, we will again be in this dilemma that whether we should use that data or we should generate our data. So the example which I have shown in the last slide is that the data which is generated from a cell line growing in 2D is actually giving some relevance to the patient prognosis, which is generated for head and neck by mostly Caucasian patients. So when we generate this data for only gingivobuccal oral cancer, our data may have a better power.
Yeah, sure. Sure. The next question is, in your research, DNA sequence is used. We can also use NGS method or not.
Yes, the data which is shown here is all developed by NGS platform. So the sequencing, the whole genome sequencing which we have shown, the DNA data is done with the NGS platform, and the whole transcriptome data which I showed uh, during the later part is all generated by the next generation sequencing platforms.
Yeah, sure. So this would be it will be a last question, and this would be a take-home message for everybody. And what is the take-home message for everyone from you today?
Okay, so take home message number one: please stop everyone to chew tobacco. To whom you see, just go spread the awareness that you are going to get very heterogeneous cancer, very aggressive. The stem cell biology is not understood, so your cancer cannot be treated by uh, the the modern biology also. So we are still working. Please stop as much as possible. And for the clinics, we have the take home message that we are coming up with the technology, with the knowledge that can be actually implemented. We can find patients who are very close to the stem cell or who are very close to differentiation state, and then we can actually segregate patients into two or three different baskets, and then maybe we should go ahead and start some kind of precision medicine approach for oral cancer, which is right now is not happening. So so these are the overall take home message. But for the students, we are showing that we have the capability of doing uh, the high throughput, high content bioinformatic studies, which has the biological meaning, which has the functional relevance. So this kind of study should be more followed up.
Yes. How do you see the research in cancer in oncology, especially in India and abroad?
Uh, yes, so we have every kind of research which is happening uh, very, very similar. I will say that people who have started to work lately, who have returned to India after getting postdoctoral training from abroad, they have got opportunity to initiate the labs. Um there are some challenges, but yes, we can overcome the challenges and we can start to have exact similar setups and opportunities which is available in abroad. uh, in abroad also. The more research on cancer should be clinic driven in India, which is right now is kind of lacking. People are still working with the same old cell lines and these things. So we need to have the focus of translation when we talk about cancer, because every day people are dying. And yeah, so obviously more than 5 people have died already.
Thank you so much, sir, for your time today with us and your sharing your knowledge with us. And thank you so much. And I especially thank you for accepting my invitation for this time too and being with us in our platform. Thank you so much.
Sure, Partha. It was pleasure, and it is always good to come back. You, whenever you feel like, you can actually ask me to come, and I will definitely come and give a talk.
Thank you
Thank you
so
very
much,
much.
sir. Thank you
Thank
so
you.
much.
Thank
Thank
you.
you. Bye bye. Thank you.